And back for part 2 of digestion. Here we go. Small little review. Stomach, duodenum, jejunum, ileum, feeding into the colon here. And so coming into the ascending colon, transverse colon, descending colon, sigmoid colon, rectum. This end portion is the cecum, and there is the little appendix. All right, so jejunum and ileum. The jejunum is about 8 feet long, and digestion happens here. So we have all these enzymes that have been pumped in from the pancreas, and this is breaking down all these proteins and carbohydrates and fats. The ileum is longer, not a whole lot longer, but it's 9.5 feet longer, and most absorption. So we've already broken everything down chemically. We've broken it down mechanically in the stomach, and now it's time to absorb. So the ileum is mostly absorption. And it's going to end in the ileocecal valve. It's the cecum, the beginning of the large intestine. Okay? Macroscopically, when we just look at it in the cadaver, for example, we can't tell where the jejunum ends and the ileum begins. So it's a little bit of a guess unless you could get a microscope and look exactly. But you know that very first portion, duodenum, what comes next is jejunum, and then you kind of guess as to where that transition would be to this other region that is more absorptive than it is digestive. This is another diagram of it. Duodenum, jejunum, ileum, and then feeding into the cecum, the ileocecal valve that I mentioned would be right there. So this valve is going to feed into the large intestine in that point. And this is why this dead end here is a problem with the appendix, because it sort of has nowhere else to go, and infections could happen if that gets stuck in there. So a little bit of histology here. The lining is folded into pleats called plica, and the surface is folded into villi, but this is easier seen with diagrams. So this is one villus, and this villus here would be, this is all the cells that line it, and then there's an artery and a vein and a lymphatic duct in the middle. We also call lacteal duct. Okay? In this, that's poking to the inside here of the small intestine. So chyme, that food that's now what we're calling chyme, is further broken down. You're going to break down proteins and carbohydrates and fats, and most absorption then is all happening in the small intestine. In what portion? In the ileum. This is just some schematic drawings for you to remember that enzymes are going to break down polypeptides into amino acids. So this is a long chain of amino acids. It's called a polypeptide. It'll be broken down via digestion, via these enzymes that are released from the pancreas. And they will now be of a size, amino acid molecules, that can be absorbed. If it's bigger than that, they're not going to be able to absorb it. We need these monomers, small molecules, to be absorbed. You can't absorb this big guy here. You will also break down starch molecules or large complex carbohydrates into sugar molecules. So we can't absorb this. We can't even absorb two together, a disaccharide. You need to have this all cleaved here by enzymes so that you can absorb one sugar alone. You know this from lactose intolerance. That means it has two. A carbohydrate with two molecules, a disaccharide, and we can't digest that, and it will go out the other end and form quite a little bit of gas. So just to remind you, one monomer, that's all we can absorb. So it needs to be broken down. The other breakdown is from fats. The fats are going to be broken down into glycerols and fatty acids, and then they can be absorbed, and this time they're going to be absorbed through the lacteal duct. And here it's illustrated for you. Get myself out of the way. The absorption is via amino acids, and sugars are going to be absorbed straight into the blood. So you imagine that absorption coming in through here and landing in these vessels here into the blood. But the fats differ. They have to first be absorbed into the lacteal. It's kind of like a little bit more careful with that. You have to kind of check to see that these are okay before it falls into circulation. Think of it that way. The large intestine comes next, or colon. That's about 4.5 feet long, and it begins with the cecum. That sort of dead-end bag is the cecum. That's where the appendix is. You will have ascending, transverse, descending, and sigmoid colon. Those are the ones I mentioned earlier. This is a nice picture. The next one, it's showing here how ileum is feeding into the cecum, and then up, across, and down. The colon then connects to the rectum. Rectum connects to the anal canal, which empties to the exterior, and that is the other end. So we've gone from mouth, from salivary glands, all the way to the anus. Rectum, and then the actual opening is the anus. Interesting here, sigmoid. And sigmoidoscopy is when the camera would come up and you would observe this area here for polyps, looking for cancer, for colon cancer, or a regular colonoscopy in which the camera comes all the way through the colon looking for polyps or precancerous cells. It has to make all those curves. It has to make the turn and then the turn here to look at all this region. Functions. We're going to complete absorption here, but mostly it's the absorption of water. So water is going to be absorbed here, salts are going to be absorbed here, and it's going to form feces. So it's sort of the last chance to regain some of that water if necessary, but no more absorption is happening in the large intestine. We want to also look then at accessory organs. You want to look at the pancreas, the liver, and the gallbladder. And the location of the pancreas, I showed you, it has to be conveniently located right by the duodenum because it's going to be pumping in its digestive enzymes into that region. And it's connected by that pancreatic duct that's going to pump things through. So it produces a juice. That pancreatic juice is full of enzymes for carbohydrates, fats, and the proteins. There it is again. It has amylase. If it's ASE, it means it is an enzyme. So pancreatic, it breaks down carbohydrates. If it's amylase, lipase breaks down fats. Protease breaks down protein. You got that whole burger down. More functions of the pancreas. Well, exocrine is what I've been mentioning, the digestion of all the nutrient groups because it has enzymes for all of them. It regulates pH also. So things are coming out of the stomach and they're coming out very low in pH. So very quickly that pancreas has to neutralize that pH and bring it closer to something like 6 or 7. So take away the pH of 2 is important. The other very important function that I have not mentioned at all is the control of sugar, blood sugar. So the control of blood sugar in your body is entirely in the hands of the pancreas because it's going to release insulin and glucagon. So those two are going to control the level of your sugars in the blood. So when blood sugar is too high, you're going to release insulin and you're going to remove some of that sugar from the blood and put it in the cells, kind of take it away. If, however, you haven't eaten you skipped a couple meals, now your blood sugar starts to drop, then what is going to be released is glucagon. Glucagon is going to do the opposite. It's going to take it from, first the liver. It's going to take it from the liver, the storage that you have, and put it into the blood so that you maintain your sugar homeostasis. Blood sugar is very well regulated, and in charge of that is the pancreas. The liver. The list is long for the liver. So it's under the diaphragm, and it's connected by a ligament called the falciform ligament, if you remember that. And here it is, beautiful liver. And functions go on and on. Produces bile, right? The gallbladder stores the bile, the liver produces the bile. And in it, it has water, bile salts, cholesterol, bilirubin. All these things are going to do what? They're going to emulsify fats. So that term is important. Emulsification. What's it mean to emulsify a fat? And I have this little drawing for you. Let's assume you have a big fat globule. You just ate this big fatty burrito. And it's got big fat globules in it. Well, it's hard for the digestive enzymes to get on the outside of that if it's very big. You want to break it up into little pieces so that the enzymes can actually chemically break things down. So emulsification just has to do with making a large fat globule into small ones and even smaller ones, right? And these tiny droplets then of fat converted from this large one is what an emulsification of a fat is. And now those enzymes can break down those fats. But at first, what you want to do is make them tiny. Other functions of the liver, you store iron. If you eat liver, you know, it tastes like iron. It stores vitamins A, D, B12. Detox, it's a detoxifier, correct? Liver problems in individuals who drink too much because it is this organ that's working overtime when it's detoxifying too much alcohol. It also has a metabolic function, and that is to create things. What it does is it converts glucose, it stores it in the form of glycogen. Don't confuse that with glucagon that I just spoke about. So we store as glycogen. And then what glucagon does is it breaks it down to be able to release it into the blood again. So for here, let's go back to this. The liver is going to make that glycogen as a storage of sugar for those emergencies. It also synthesizes plasma proteins, so proteins in the plasma that are, for example, for blood clotting. All those are going to be a function of the liver. The gallbladder, I've told you, it is going to store bile, concentrate and store bile. Now, some individuals don't have a gallbladder because it's been removed. Why has it been removed? Because they had stones. Why did they have stones? Hard to tell exactly why, but it comes from the crystallization of cholesterol. So maybe slightly dehydrated, the cholesterol that should have been in there crystallizes and it forms stones. Passing those stones is very painful. So individuals who have too many stones and need to remove that or remove it, the liver continues to make bile, but you don't have a storage unit for it. Okay? And they pretty much digest pretty well still. So there's the gallbladder end of the liver, very green, and it's got its duct here that's also going to come into, guess where? The duodenum. This is bringing bile from the liver into the storage unit, which is the gallbladder, and it will go back out again. Let's look at some clinical conditions. Peptic ulcers, they're gastric or the duodenal mucosa from the duodenum, can be eroded by stomach acid. We have a thick layer of mucus there, but in the event that that mucus is worn off, and we do have evidence that bacteria are able to do that, they're able to eat this away. And then with a lack of that coating, you have that erosion of those cells forming ulcers. Gallstones, I mentioned, they're crystals of cholesterol. I'd like to end on this worms. Worms are interesting. We have pinworm in this county. There's a lot of worms around the world, but we don't have a whole lot of them. This is one that is present around here in little kids. Little kids pick this up at the playground. They have everything in their mouth. And you have tiny little worms that they develop. The female worm migrates to the anus. This is very lovely to end on this. And it causes itching, so the little kid would be itching their anus and collecting more eggs and passing those eggs on to anyone else who touches the little hands of the little kids. I'll show you a picture of it. These are with a ruler here for you to see the size.